Good evening, and thank you for joining us for tonight's Woo You event with Dr. Natasha Heineman. The title of her presentation for tonight is Clinical Trials on Presbyopia Eye Drops: How to Compare Apples and Oranges. I'll be your host tonight. My name is Dr. Ariel Sorenzi. And a big thank you to Lens Therapeutics for exhibiting at this event. This will be worth 1 hour of CE credit, so please be sure to attend for a minimum of 50 minutes. For your COPE certificate, just please fill out the survey link that is in the chat now, and it'll also be there when the webinar ends. Your CE certificates will be delivered via email and sent to ARBO with the OE tracker numbers provided. But we'll also have a QR code at the end if you want to use your OE tracker app and just get that credit instantly through your phone. Otherwise, CE certificates will be emailed within about 4 weeks. We will save questions at the end of the presentation, so you can ask those either in the chat or the Q&A area there at the bottom. If you're on your phone, you can find the Q&A at the top or the chat function under those three dots titled More. So I'd like to introduce our speaker tonight, Tasha Heineman. Dr. Tasha Heineman has been practicing optometry since 2003. She graduated magna cum laude with BS in biochemistry at Roberts Wesleyan College in Rochester, New York, where she also played basketball, captaining her team. She then attended Pennsylvania College of Optometry, where she received her BS in visual science as well as her doctorate in optometry. At Scott and Christie Eye Care Associates, Dr. Heineman serves as a director of clinical research for their Eye Research Institute. In this role, she serves to effectively advance the future technology of eye care through clinical trials with a focus on dry eye, glaucoma, blepharitis, presbyopia, myopia, and allergies. In her spare time, Tasha enjoys hiking, live music, sports, and spending time with her family, her husband Tom, and her young daughters, Joy and Claire. So we are so excited to hear from you tonight. So thank you so much for sharing all of your expertise and knowledge with us.
Well, thank you so much for having me tonight. Thanks everybody for joining. I'm gonna ask a couple of favors, perhaps I'll say. I live in Pittsburgh, which is the wintry Northeast, which means that there are germs everywhere. I have two small children who bring home all of the germs. I have a germ, so I'm gonna actually turn my camera off because I'm gonna be coughing and wiping my nose, and I don't know that any of you really want to see that. So that's one thing. Bear with me while I deal with all that. I do have these financial disclosures, working with Lens Therapeutics and Tarsus Pharmaceuticals, and for some reason, there we go. And they've all been mitigated. So tonight we're going to talk about clinical trials on presbyopia drops, how to compare apples and oranges. So like you just heard, I have been practicing optometry since 2003. I graduated and from the school in Philadelphia and moved shortly thereafter to New York, starting as many of us do, I think, in retail optometry. I worked for a big corporation in retail optometry and moved with them to Pittsburgh about 4 years after graduating so I could be closer to family. Stayed with them for a couple more years, moved then to a small private optometric practice for a few years. And then in January of 2017, started at the practice I'm at now, which is an MD-OD practice. Interestingly, they saw on my resume that I had research experience. I say that very loosely because it is definitely not clinical trial research. I did a biochemistry research summer studying the genome of chlamydia. It was PCR and biochemistry and genetic stuff, nothing at all like clinical research, but this practice wanted to grow their clinical research program and thought that I might have the ability to do that. Not something that I ever knew existed in optometry, wasn't an area that I really understood very well, but I have grown to absolutely love it. I love what I do. It's very different. It's interesting. It's different every single day. When I started, I was doing 1 to 2 days a week in general trials, and then the rest of the time I was doing general optometry. We started with 2 to 3 to 4 trials at any given time. But since then, we've grown tremendously. So now I'm full-time research. It's all I do. We have 25 to 30 clinical trials at any given time. We do all 4 phases of trials, 1, 2, 3, and 4, for both devices and pharmaceuticals. We do blepharitis, dry eye, presbyopia, myopia, glaucoma, IOLs, viscoelastic, allergy, MGD. I mean, anything anterior segment. We don't do anything posterior. We don't have a retinal specialist. And since then, you can see these numbers here. We've done 143 clinical trials, over 3,500 patients. 17 of those trials were specifically for presbyopia and over 300 patients in those trials. 13 of the medications or devices that we've worked on have made it to market, and there are 3 more waiting for FDA approval. So here is, I don't know how well you can see this, but basically this is in our waiting area in our office. It's showing the things that we've worked on that have come to market. It's great for patients to feel a little more at ease that, the thing that they are participating in may actually come to fruition. It's great when we have drug companies come to visit our practice, that they can see that they might get a spot on our wall. On that wall, you'll see, maybe if you can see, Mibo, Xdemvy, Closey, Vuity, Vivi, iDose, Isuvis, Tyrvaya, Durista, Xiidra, Rocklatan, Repressa, Vyzulta, pretty much anything anterior seg that has come to market in the last 10 years or so, we've participated in, which is also why I love this, because it kind of gets us ahead of the curve about what's coming to market. Tonight, we're going to go over 3 different things. The basic what's involved in conducting clinical trials, the landscape of presbyopia drops, what's currently approved and what is now in development, and then how do they compare, and basically how to review clinical trial outcomes. So when you have a sales rep come in and they hand you material like they love to do, what does it even mean? I actually saw one of my favorite reps today. I've known her for years. And she handed me some information, and because she knew I was doing this, and I said, well, here's what I'm going to tell people to look at when they look at your stuff. I won't say which company she's with, but their job is to sell. It's our job to understand what it is that they're showing us and what actually makes sense. So first, let's talk about a general idea of clinical trials. So like I said, when I first started, we didn't have 25 or 30. We had a couple. It's now full-time because of how many trials that we do. And what I'm producing is data. So my job is to get high enrollment of patients and to provide quality data of the clinical trial. This is going to require facility and staff. When I first started, it was just me. I didn't have anybody else. So I was doing everything. I was doing the paperwork part, which there is a lot of that. I was seeing the patients. I was entering the data into the computer system. And now we have 4 full-time staff members so that I can focus really on just the doctor interactions. But it does take some time to get to that. The most important thing too is growing the relationships that's with the patients and with industry. One of the first things that I did was build a database of patients, because typically when a patient does one study, they enjoy it. They enjoy the boutique experience, their one-on-one interactions with myself and my team, who are fantastic, by the way. And then they want to do more. So if I get a study that's for presbyopia, I've got a list of those 300 patients that have already done a presbyopia trial and call them, and they want to do it again. Same thing with industry. It's a small industry. So somebody that I worked with on a Vuity trial back in 2019 called a year ago. She's with a completely different company in Australia and wanting us to do a glaucoma trial. So building the relationships is very important. And then meeting those goals, meeting the enrollment goals. When you start a trial, they ask you, how many patients can you put into this trial? If you say 30 and you give them 2, you're probably not going to get a call again. So it's important to understand the study as well as what you have in your patient database, and to see if you can actually meet those numbers. It's also important that If you don't have the patience, don't say yes. It's okay to turn down a study if it's not something that's in your wheelhouse. I get asked frequently to do neurotrophic keratitis. We don't see much of that, so it doesn't make sense for us to do that. And always I hear, thank you for being honest so that we aren't wasting anyone's time. So I want to go over the key players, the people who you're going to interact with when you're doing studies. The first is the sponsor. The sponsor is the drug company who has developed the new medication or device that is being tested. They pay for the conduct of the trial, and their main roles, the people that you'd interact with, are the clinical operations lead and the medical director. In order to pay for the trial, sometimes they need funding. So the sponsors are all over the place, from huge companies you've all heard of, Bausch and Lomb, Alcon, AbbVie, et cetera. But we also work with a lot of small bio startups who they have one drug, that's their baby. If that's who I'm working with, I'm going to have a lot more interaction with the sponsor than if it's a big company like Alcon, because they have lots of products. They have a whole dedicated research and development department. But these smaller companies, you get a more interaction with. A great example of that is Tarsus. I know we all know who they are now, but 5 years ago, nobody knew who they were. So now with XDEMVY, they're big, and we have less interaction with them now than we did before. Next is called a contract research organization, which is a CRO. Basically, they're like a liaison between the sponsor, the drug company, and the site, which is us. They oversee the conduct of the trial, and they're responsible for collecting the data from the sites to be shared with the sponsor. So they have a project manager, a data manager, a monitor, who we also call a CRA. Sometimes those roles are contracted out in bigger companies, but in smaller ones, they're going to stay in-house. They also help with protocol writing. They select the sites that are going to be involved in the trial, and they're in charge of the data. It's called EDC, where we enter the data so that the sponsor can see the information. There's something called an institutional review board. So this is an independent group of administrators whose whole job is to review and monitor studies with human subjects. So they're there to protect the rights, the safety, and the welfare of the enrolled patients. They're a contact for patients if there is a concern about the conduct of the trial. You don't ever really want to get a call from the IRB, because if a patient called, that suggests there's a problem, but that's their role. They're making sure everything is done ethically and the right way by means of the patient. They're registered with the FDA, and they have specific roles that have to be filled. So, for example, there has to be a person that has a scientific background. So that would be like a physician or a scientist. There has to be someone who has a non-scientific background. That could be a teacher. And none of these people can have conflicting interests with the study being tested. So there's a central IRB, which is a large group that can conduct the study across the country. There are also local IRBs. So those are more likely to be found in hospitals or like a big educational system like Duke. They use their own local IRB. So again, their role is to protect the patients. The site, that's us. So that could be a practice, it could be a hospital, it could be a research facility. Our job as a site is to enroll patients and execute the protocol. And again, my product that I am delivering is data as well as enrollment goals. The roles here are principal investigator, a sub-investigator, a study coordinator, and a clinical research assistant. The principal investigator has to be a physician. It could be an optometrist, but it has to be under the scope of licensure. So, for example, I can be a principal investigator on a presbyopia study because the medication that will come to market is something I could write a script for. I could not be a principal investigator as an optometrist for a surgical device because I cannot myself do surgery. So in those cases, the principal investigator would be an ophthalmologist, and I would be the sub-investigator, who is another physician that does the assessments with the patients. The study coordinator, their job is to do all of the daily tasks, all of the paperwork, make sure that things are being done according to the protocol. And then the clinical research assistants are the people that are doing the testing. So things like checking visual acuities, scribing for the doctor, etc. The principal investigator, they are responsible for the entire conduct of the trial. So from the very beginning, the first document that they fill out is called a 1572. This goes to the FDA that explains who is in charge of the study and who else is working on that study. And basically, it means that they're responsible for everything that happens, whether they've done that or not. So the principal investigator has to trust those people underneath them are doing things appropriately to make sure that their license is protected. Because ultimately, if the FDA decides that they're going to inspect the site, no matter who may have quote unquote screwed up, it's the principal investigator whose name is on that 1572 and who is responsible for the product that was delivered, which is the data. Next is the FDA. We all know about them, the Food and Drug Administration. Their job is to oversee conduct of the human subject trials to make sure they're conducted safely and in accordance with the federal laws. They review the data when the data is presented to them by the sponsor to make sure that whatever the new medication or device is safe and effective. And then after that, they can and often do inspect the sites, which would be us. We have been fortunate enough to have 3 FDA inspections in the last 18 months. That's sarcasm. I don't know how fortunate it felt at the time. All 3 of those inspections were great. We had no issues, but they do come and they want to make sure that the data that we gave is correct. So the data that the sponsor gave to them matches the data that we gave the sponsor, and that it wasn't made up. Unfortunately, I think that it has happened in the past. I've had the FDA inspector tell me that, they were happy to see charts for our patients, that they were actual real patients. They also can inspect the CRO, the Contract Research Organization. So if they go to a site and find a problem, they may find that the problem came from higher up, and they need to make sure that it was handled appropriately at that level. And they can even go to the sponsor if they feel that that's necessary. If they do agree to approve a medication, then that medication will come to market. If they don't, they often will say you need to go back and do an additional trial to prove this, or we don't like this data point, you need to do something again. They also have to approve the protocol itself prior to it going out into the clinical research sites. And they also set the primary endpoints, which we'll talk about later when we talk about the presbyopia trials. Their role is to tell the sponsors what they must prove. There's 3 abbreviations that we talk about a lot with clinical trials in the FDA. One is called an IND. An IND is an investigational new drug. So a sponsor will have to file this with the FDA when they would like to start a trial because they have a medication or device that they would like to get approved. Then once they've gone through all the trials and feel they've proven the data points that they need to, they will submit an NDA, which is a new drug application. After the FDA receives that, the FDA then has to give what's called a PDUFA date. I don't know what that even stands for, but basically it's the date by which the FDA has to respond to the NDA, and it's usually about a year. So a clinical trial from conception to FDA approval can take 7 to 15 years. I mean, it can take a very long time to go through the process, which is why when something new comes out, it's exciting because it's been through so much testing. Excuse me, there was my first cough. Okay, so next, what are the elements of clinical studies? So the most important thing is the protocol. This is basically a playbook. This tells us what we are doing, when we are doing it, in what order we are doing it, and we have to adhere to the protocol to the letter. That is what the FDA is looking for. They've approved it already. They've already told the sponsor, your protocol is approved to be implemented. Now we have to implement it to the letter. First, they have a hypothesis, and we know what a hypothesis is from, science in high school. It is a proposed prediction, what they are expecting to see. a primary, secondary safety endpoint. The endpoints are specific measurable outcomes. The primary endpoint is set by the FDA. The secondary endpoint is typically set by the sponsor. Safety endpoints are there for the safety of the patients. There's also exploratory endpoints. So examples of these things would be in cancer research. A primary endpoint might be the overall survival rate of a patient, whereas a secondary or exploratory endpoint, it may not be statistically powered, but it could generate new hypotheses for future trials. There's inclusion-exclusion criteria. These tell us what types of patients are allowed to be enrolled and what types of patients are not allowed to be enrolled. There's a schedule of assessments, which again is going to tell us exactly what assessments need to be done and in what order. So taking a medical history, checking a vision, doing a slit lamp exam, checking a pressure, and in that specific order. The investigational product, that is going to be either the pharmaceutical or the device, whatever is being tested. And it has to be stored very specifically. It has to be in a secure location and very frequently with temperature monitoring. So when we talk about starting to do clinical trials, these are all things that you have to have. You have to have a location where you can lock a cabinet inside of a room with a locked door, inside of a suite or a building with a locked door, because these investigational products are worth potentially millions of dollars to the sponsor. So they need it to be very secure. Also, documentation. It's extremely important. If you don't like paperwork, don't do clinical trials because there is a lot of it. We have to document adverse events. Now, adverse events are basically side effects, but in the world of clinical trials, we call them adverse events or AEs. Anything that happens to a patient from the time they sign an informed consent to participate to the exit of the study is considered an adverse event. So whatever I have right now, cold or whatever, that's an adverse event. Did my eye drop cause it? Obviously not, but we still have to report that that patient had this adverse event. Protocol deviations are expected and they're okay. When I first started, I got myself really worked up when I would make a mistake and have a protocol deviation, but it's real world. The FDA expects them. You don't want to have big ones where you put a patient in a study that should not have been, but you might have done an assessment out of order based on need of the patient. These things all have to be documented, reported to the sponsor, and then reported to the FDA so that they can ensure that you are aware that the mistake was made and what you did to correct that mistake. The documentation has to follow what's called ALCOA, which is attributable. You have to show who took the information, legible for obvious reasons, contemporaneous, which means it was done in real time. So in the real world, you might say, oh yeah, my patient an hour ago, I checked the pressures, they were 15, and then you write it down. This has to be done in real time. So you can't try to remember what a pressure was. You must write it down then. It has to be original. So no whiteout, no erasing. Everything has to be original as well as accurate. So documentation is extremely important because, again, I've said it twice, my product is data. So that data has to be good. So what makes a well-designed clinical study? Most importantly is the protocol. The protocol has to make sense. If I read it and I don't understand it or I have a lot of questions, the FDA is going to also have questions, and that's not a good start. You want to have inclusion, exclusion criteria that are real world. Sometimes they'll put something in there that doesn't really make sense, excluding a diabetic for no reason other than it might confound the data. Ease of patient participation. Are you going to be able to find patients who want to do this? Does the protocol exclude or prohibit your standard of care? This I'll see in IOL studies where they'll say you can't perform a femto or an LRI. My ophthalmologists do a lot of femtos and LRIs. So if we're not allowed to do that in the scope of the trial, it's going to be hard to find patients that could participate. And then finally, what is the control or vehicle? So whenever there's a clinical trial, you're comparing it to something. You're comparing it to a control or a vehicle. what is it? And is it something that makes sense? And how do you eliminate people that might respond just to a vehicle? That's important in dry eye studies. The comparator is usually going to be a vehicle, and the vehicle, I explain to patients, is like driving the car. The driver is the medication, the vehicle is what drives it in. So in a dry eye study, often it's just basically the drop without the medication that's the vehicle, and you don't want them to have a placebo effect. So how are they eliminating those patients? Common challenges to conducting clinical studies is kind of the opposite of what we just said. So protocols that don't make sense, contradictory, things where it says on page 2 to do a pressure before a slit lamp, and on page 10 it says to do the slit lamp before the pressure. Which one's which? The FDA wants me to follow it, but it doesn't make sense. Inclusion, exclusion criteria that are prohibitive. So you're going to eliminate half of the patient population with these very prohibitive criteria. If it's complicated for the patients and my staff, if my staff decides that this is too hard, it's going to be hard to convince them to help enroll patients. And sometimes there's required equipment that's not standard, and sometimes that they do provide that, but sometimes they don't. So that's another thing that can make it difficult to conduct a trial. So how do you recognize when you are being handed this material from a sales rep? How do you recognize a poorly designed study? And how do you recognize if they're trying to pull the wool over your eyes with the results? So review those inclusion exclusion criteria. They may not give you the full list, but they'll give you highlights. Does this sound like a typical patient, or does this sound like a needle in a haystack? It's hard to know if it's going to work in the real world if they're pulling needles out of haystacks. Was the highlighted endpoint an easy target? So if it's made its market, it obviously had to meet the primary endpoint, or the FDA would not have approved it. But are they talking to you about the primary endpoint, or are they talking to you about secondary or exploratory endpoints? They have to say, and that's important because something could meet a secondary endpoint with a much higher number than it met the primary endpoint. That also leads to the statistically significant verbiage that they love to use, which is important, but it's not always real world. If you can't really understand a graph that they're showing you, it's probably because their biostatisticians did a wonderful job of, I don't want to say manipulating the data, but making the data work for them. And then also, what are the side effects? What were the most commonly reported adverse events over the course of the trial? Because if they happened in the trial, they're very likely going to happen in the real world. And is that something that you want to expose your patients to? So again, I think most important here is this whole statistically significant. Remember, they met an endpoint, but the endpoint may not actually be that great in the real world. Okay, so let's move into the world of presbyopia. What's approved? What's in development? Right now, there's two basic strategies from a pharmaceutical perspective in treating presbyopia. First is lens modulation. The idea here is to change the elasticity and the flexibility of the lens to accommodate and allow for near vision. The other idea is meiosis, which is basically like pinhole effect of a camera, where we reduce the pupil to expand the depth of focus to bring near vision into focus without impacting distance vision. So you can do that in one of two ways, by either stimulating the iris sphincter muscle or inhibiting the iris dilator muscle. So busy slide, but basically what this is telling you is that with lens modulation, it has not been successful. So it's fascinating. I really love the idea of it, but the trials that we've done with this have not yielded the results that the sponsors have hoped for. Basically, it's been ineffective to date. I think, to me, the most interesting question, nobody's been able to answer it for me because they can't yet. What happens when a patient who has been on this drop of, if there were to be one approved, when they need cataract surgery? Is it more complicated? Has the lens material changed so much that it's harder to extract the lens? Is it easier? I don't know, just something to think about. But again, at this point, this has not been successful in clinical trials. The method that has been successful is with the miotics. So there's two basic types of miotics. There's the type that will stimulate the iris sphincter. These are the parasympathomimetics, such as pilocarpine, carbachol, and aceclidine. And then there's the ones that will inhibit the iris dilator, which are the sympathomimetics, brimonidine, and phentolamine. So if we look at the iris sphincter medications, pilocarpine and carbachol are considered non-selective, which means that they are acting on the pupillary, the iris sphincter, as well as the ciliary body. And then there's aceclidine, which has been shown to be selective, and it is only affecting the iris sphincter. Phentolamine and brimonidine are the two iris dilating medications. Phentolamine has actually been approved for reversal of mydriasis, which is Ryzumvi. And they've also continued to work on it for the purpose of treatment of presbyopia. So if we're going to focus first on the iris sphincter medications, you have to understand what's going to happen as a potential side effect. So when these medications stimulate the ciliary muscle, you get two unintended consequences. You get a myopic shift, which will induce myopia, which obviously will negatively impact your distance vision. And you could also get a ciliary spasm headache, which is, as that ciliary body is stimulated, it's causing a brow ache that can be very bothersome to patients. We saw a lot of that with Vuity when it came to market. And then the other thing is that it can displace the lens anteriorly and thicken that, which can then lead to that vitreoretinal traction, which can then lead to the retinal tears, breaks, and detachments, as well as the potential for posterior synechia. So a goal would be to have this pupillary miosis without stimulating the ciliary body and therefore getting these unwanted side effects. So there's been independent research that has shown how these three medications, the three iris sphincter medications, affect the ciliary body. And basically what this is showing is that aceclidine requires a lot more drug to get the response of the ciliary body. That's how they've determined that this medication, the aceclidine, has pupil sphincter selectivity, and how the other two are non-selective because they're affecting both the iris sphincter as well as the ciliary body. And here is the other unintended side effect of the myopic shift. So when they've done this independent research, they've found that the ideal pupil size for getting a near improvement is what we call a sub 2 millimeter pupil. All of these medications are capable of doing that, as you can see here. So 2% aceclidine goes to 1.4, 2% pilocarpine goes to 1.35, 3% carbachol 1.63. But look at the other side at the induced myopia. Hardly any with the aceclidine, but that a diopter plus of induced myopia is a significant amount that will really detract a patient's experience. And so what they have to do is decrease the concentration of the medication in order to mitigate that higher myopic shift. So here's what we have in the works. So Vuity is pilocarpine 1.25%. This was approved October of 21. Closey, which is pilocarpine 0.4%, which was approved in October of 23. Brimacol, which is a combination of carbachol and brimonidine. This is currently they're ending their phase 3 trials. They're expecting and hoping that it will be commercially available in 2026. Nixol, which is phentolamine, that's one of the iris dilator medications. They are in phase 3 studies now. Microline MAP, which was pilocarpine 2% spray, was actually discontinued because they were not getting the results that they wanted. And then Lens 100, which is aceclidine. This was submitted the NDA, the new drug application, to the FDA in August of 24, with a PDUFA date, if you remember that, expected in August of this year. So we did all of these studies. I'm very familiar with each one of these medications and how they worked in our practice. So let's compare the actual trial outcomes. So Vuity, which we all know from Allergan, those trials were done in 2018 and 2019. There were Gemini and Virgo. Closey, which is expected to be coming to market very soon, was a smaller bio startup company called Orisis. They did the NEAR 1 and 2 trials as well as a safety trial. Those were in 2020 to 2022. Brimacol is, like I said, just wrapping up their studies now. Nixol did the Vega trials and are still in trials. And then Lens 100 by Lens Therapeutics did Clarity 1 and 2, which were for efficacy, and Clarity 3, which was a safety trial. So remember what I said before, the FDA sets the endpoints that must be proven through the protocol in the clinical trial. So for presbyopia, the primary endpoint for all of them, they're all the same, is that there must be a greater than or equal to 3-line improvement in distance corrected near visual acuity. That's 15 letters. So it's not a 3 full lines. It's just in clinical trials, we measure things by letters, not by lines. So 15 letters of improvement. At the same time, there can be no loss of distance corrected acuity of more than 5 letters. One of the things that we'll talk about when we go through each one of these trials is that it's important to look at if they did monocular or binocular acuity reporting. The secondary endpoint, which is usually determined by the sponsor, the drug company, varies. They could look like different things. It could be greater than or equal to 2-line improvement in the near vision. It could be a % of subjects achieving 20/40 or more. It just depends on what they think their medication can do or what they've seen it do in previous trials. So Vuity was the first one that came to market. It was a big deal. It was a big deal for us to be a part of the trials. And it was a big deal for the industry because it was the first time we had anything in our arsenal outside of contact lenses, things like that. So this was 1.25% pilocarpine with a BAK preservative indicated for the treatment of presbyopia in adults. Their phase 3 trial, here's your inclusion-exclusion criteria that we talked about. The ages was 40 to 55 with a spherical equivalent refractive error of -4 to +1 and no pseudofakes. And they reported binocular acuity outcomes. So here's what they've published as far as their results. Their primary endpoint, which again, set by the FDA, was at 3 hours post-dose. And they found that 31% of the participants responded to that primary endpoint in Gemini 1 and 26% of the patients in Gemini 2. So what are we looking at here? We got a limited age range, right? So these are your early emerging presbyopes. You have a decent range of spherical equivalent, no pseudofakes allowed, and binocular acuities. So binocular acuities, we all know you get 2 eyes, you see better. So what would you expect if you were looking at monocular? You'd expect an even lower amount. So they proved their endpoint. They got statistically significant results, but 31%, 26%, not that great of a response if you're looking at real world numbers. They even then found that because the duration was so short, they had to go back and do a BID dosage study to see if we dose twice a day, if we'd get a better outcome. So they added a second drop at 6 hours. And then their primary endpoint was day 14 at the 9th hour. And there they found 35.1% had that 3-line improvement. So they met the primary endpoint, but it's a low number of subjects showed that you had limited efficacy and a short duration of effect. So on each one of these trials, I'm just going to show you a couple of graphs. And again, these are all from their own marketing material. A lot going on, but I want you to look at the pupil size. Remember, we talked about the sub 2 millimeter pupil. So if that's the bottom left graph, you'll see that they got to a little under 2 perhaps on the Virgo trial. at the 1 hour mark. And that is when they had their greatest efficacy. That's going to be the graph to your right. So again and again, you're seeing the smaller the pupil size, the better their vision's going to be. So Closey was approved in October of 2023. It is not yet out. It is not yet commercialized. This is an example of a smaller bio startup that needed to get funding to get the drug to market. So I expect it to be out probably in March or April of this year. They're also pilocarpine, so 0.4%, a much lower concentration. It's also preservative free. Their big thing is that the neutral pH improved the corneal penetration, which they believe, and their results would show, has better efficacy with lower side effects. So here's your population, your inclusion-exclusion criteria, 45 to 64. So they've broadened the age range a little bit with a wider spherical equivalent range, minus 4.5 to plus 2. And here they found 39% and 48% of patients had that FDA required greater than or equal to 3-line improvement. That endpoint was at 1 hour post-dose 1 and 1 hour post-dose 2. So this is approved for QD or BID dosing. The second dose would be 2 to 3 hours after the first. Their maximum response was 55%. So the best they found was 55% of the patients met that 3-line improvement at post-dose 2 on day 15. And they did monocular acuity. So one could expect that if they were looking binocularly, you might get 60 to 70% of people with that improvement, but they did it monocularly, which I think it shows that there is better efficacy than you might see on these paper. So here's the same type of graphs that we looked at for the Vuity. The size of the pupil is on the bottom right. Never got below the sub 2, but they did get close at 1 hour post-dose 2 and 1 hour post-dose 1. That is where you'll also see the best efficacy, the highest percentage of patients that got that 3-line improvement. So again, that independent research talking about the sub 2 pupil, when you see the data, you will see that the best efficacy is when they have the smallest pupil size. So Brimacol, this is the one that is wrapping up now with they're hoping to have this out in market in 2026. It is a combination of carbachol, which is a non-selective iris sphincter agent, in combination with brimonidine, which is the iris dilator type. The idea here was that the brimonidine would inhibit contraction of the ciliary muscle, which they hope will mitigate the side effects that can occur with those miotics. They had emmetropic patients. Their definition of emmetrope, by the way, was a spherical equivalent of plus or minus 50 with no more cylinder than half a diopter. They also expanded their age range, so 45 to 80, and they did include pseudophakics. They did achieve primary endpoint in the Brio 1 trials all the way out to 6 hours out of the 10 that they tested. And they reported binocular. Again, to me, I think binocular is going to be better because you can extrapolate out what the vision could be binocularly. So here again, we see the peak of the responder rate. They did, their Brimacol is the first bar, and then they separated out the components. And indeed, it does show that the combination of the carbachol and the brimonidine does give a better result than each of those individually on their own. Here's pupil size. You will see they are not quite under 2. Their best result was with, again, the combination. And that is, again, where they had the best efficacy, was in that 1 to 2 hour mark. Nixol is the phentolamine, which is an iris inhibitor, an iris dilator inhibitor. Their original study, they had Nixol at night with pilocarpine in the morning, and their results are here. At that 3-line improvement was measured at 1 hour with a 61% responder rate. This again was reporting binocular acuity. They're currently working on their Vega 2 study data, and here they decided to eliminate the Pilo and only use the Nixol. One would assume because of the potential side effects of Pilo, the interaction with the ciliary body. But if you look at the results that they found from the Vega 1, the highest bar is the Nixol plus the low dose pilocarpine. That's what LDP stands for. So that's where they're getting 61%. That's pretty good, right? 61% of your patients had an improvement, but they've decided to scrap the LDP, and that's what this circled bar is. That is just the Nixol. So way less efficacy, but again, I think that the idea was to eliminate the risk of adding that pilocarpine. Here's your pupil size. So we got down, the lowest you'll see is 2.1. That was again with the Nixol and the low dose pilocarpine. That is again where the best efficacy was found at that 1 hour mark. If you go up to the other blue line, there's 3.2 is the lowest they got there. That is just the Nixol. So I think they're going to end up having to do a little bit of reformulation, because a 3.2 millimeter pupil it provided what, 30% back here, as opposed to the 61% when we were down to the 2.1 size pupil. And then there's the lens 100. So this is aceclidine. Again, this is the only one that is considered pupil selective. So has minimal to no impact on the ciliary body. It was single dosing with an extended duration of action. And their big thing was, we're going to hit this sub 2 millimeter pupil. And they wanted to do it all the way out to 10 hours. The drop was preservative free. Aceclidine has been around for decades. It actually made enrolling patients in this study a little bit easier, because the patients recognized that it wasn't a brand new product. The safety profile was known, because it's been used in Europe for glaucoma since the 70s. Here, we never had it approved for glaucoma or anything else, but this is where this company decided to work on it for the pupillary miosis and presbyopia. So they did the Clarity 1 and 2 efficacy trials, as well as Clarity 3, which was a safety trial, and again, reported monocular visual acuity outcomes. They submitted to the FDA in August, so we're expecting to see approval later this year. So here's their study design. So we've got patients aged 45 to 75, again, a higher range of patients with a pretty typical at this point, I would say, a refractive range of -4 to +1. They did allow post-LASIK patients as well as pseudophakics. And this to me was interesting, that they required the near vision to be at least 20/50. And we'll come back to that, because that does matter. So here's their results. I mean, I think that the first thing I look at when I see this is 91, 95, 91, 87. And this medication seems to have worked the best of those that we worked on in trials, but this is still an example of how they're going to show you numbers to get your eyes to draw to it. So the color orange, if you look at the graph, is two lines or better. That's not the endpoint that the FDA requires. That would have been a secondary endpoint. The primary endpoint was the dark blue with a 3-line or better improvement. But look, it's still 71%, 73%. I mean, this is a significantly better results than you've seen in any of those other medications. It also goes all the way out to 10 hours at 40%. So you probably don't remember these numbers, because why would you? But Vuity, their top line results was about 30%. Closey was about 45%, and that's at 1 hour. This is at 10 hours. Brimacol was at 50%. Nixol was at 47%. So this is a much higher result than we've seen in the other studies. So this is going to show you that it's repeatable. So at day 1, at day 15, and at day 28, at all these varying time points, there was this significant improvement with the 3-line or greater improvement in near vision. In addition to that, it's not like they stop at 3. They also are checking how much better than 3 lines do we get. So 84% of the patients had 4-line or better improvement at some point over the trial. 52% had 5 lines. 24% had 6 lines. This again is why I said we'll come back to this, where they had to be at least 20/50 to enroll in the study, 20/50 at near, because they had a lot more room to improve. You can see how much some of these patients were able to see an improvement in their near vision. So it makes sense that you would expect to see the patients that had more presbyopia have a bigger improvement in vision. So those who had advanced presbyopia, here defined as being worse than 20/80, those had a higher improvement in the greater lines of near vision. So this was actually, I think, an exciting find. The FDA requires that you don't have a decrease of 1 line or more in distance. And so that has to be proven. But what Lens found in doing this study is that they actually had an improvement in distance vision. So at the 3-hour endpoint, 39% of the patients actually improved at least 5 letters in distance. And again, I think what that's showing is that sub 2 millimeter pupil is going to improve not only near, but the distance. And I guess, it makes sense. If there's no ciliary body interaction, there wouldn't be that myopic shift. So it's again proving that there is a lens selectivity. I also told you before, you need to look at what the side effects were for lens. That is the study that we did. We did the safety study. So our efficacy results were strictly anecdotal, and the patients liked the drop. The side effects were very, very mild. So adverse events in clinical trials are defined by if they need treatment, they're automatically considered a moderate side effect. So these are all mild because the patients did not seek out treatment for the fact that it burned when they put them in, that there was some dimness from the constricting of the pupil, that their eyes turned a little red, and that there were some headaches, again, because of the decrease in size of the pupil. None of these were considered serious, and very few people dropped out. And honestly, I think those people dropped out for other reasons. Adverse events are a reason that people drop out for sure. But they are also, we're not allowed to, actually, I shouldn't say that we must ask them if they have any side effects. So when a patient comes in, the first thing we say is, do you have any changes to your medications or any issues with the drops? We are required to elicit if they're having any side effects, because it's important for you to know as treating physicians what your patients may expect from using the drops. So these were all very mild, basically sight irritation and redness. So we're going back to that sub 2 millimeter pupil. 99% of the patients in this study had a sub 2 millimeter pupil, at least at some time point. And the lower the size of the pupil, the greater the efficacy, which we saw when we looked at all of the results of these trials. So these patients, if they had less than a 1.5 millimeter pupil, there was 78% of them had that near 3 line of near vision improvement. Again, same thing, distance. All this improvement in distance was also because of that sub 2 millimeter pupil. We don't talk about, I said this before, but we don't talk about 20/20, 20/30. We do logmar, which is more defined by letters. So when you see minus 0.06, that equates to 3 letters. So when the pupil size was 1.5 to 1.75, those patients had an average of 3 letters of improvement in distance vision. Okay, so when you're looking at these studies and you're being handed these brochures from your sales reps, try to remember the things that we talked about. So efficacy with presbyopia drops, did it improve their near vision? Did they hit that sub 2 millimeter pupil? And can you correlate where the 2 millimeter pupil or the smallest size pupil came in with the efficacy? What's the duration of action? Is it, was their endpoint at 1 hour? So a patient uses a drop and an hour later they can see, but 2 hours later, maybe not so much. Did they need more than one dose to get to that improvement? And then what are the undesirable side effects? Are they minor, like that irritation upon instilling the drop, or were there major side effects, things like retinal detachments or retinal issues?